Okay, so hi everyone, I'm Hania Nizami, and I'm going to be teaching you guys glycosaminoglycans, proteoglycans, and glycoproteins. Just so you guys have an idea of how I construct these, I try to make these as interactive as possible. So usually what I do is that in one segment, I'll teach you the concept, and then the next segment, I'll quiz you on it. So if you guys could just answer my quiz questions in the chat, or you can unmute yourself and answer them, that'd also be good. And if you guys have any questions throughout, you can just type them in the chat box, or again, unmute yourself and I'll answer them as we're going. But I will have some time at the end to also answer them. But I'm just saying that I do like to answer questions as I'm going through it because I want to make sure that everyone's keeping up. So let's start. This lecture is pretty easy, so I think it'll be short. So pretty much, let's just go over an overview of what glycosaminoglycans are and why we're talking about them. Pretty much is that the reason that we care about them is because we have this thing over here, which is called an extracellular matrix. And an extracellular matrix is pretty much the area outside of my cell. So for example, if this are a bunch of cells, these are my blood vessels, you can notice that the space in between the two is called the extracellular matrix. And so any communication from the blood to your cells, or from your cells to your blood, is going to have to go through the extracellular matrix. And this extracellular matrix is made of what we call glycosaminoglycans, which are pretty much modified sugars. And we're going to talk about how they've been modified in a little bit. And they also have some other stuff like collagens, laminins, and that kind of stuff. But the one that we're obviously going to be focusing on are the glycosaminoglycans. Okay, so the main concept that I want you to know about glycosaminoglycans is that, like I said, they're modified sugar. So that's why they end with glycans, because glycans mean sugar. And they're modified specifically in a way to be very highly negatively charged. And this is really important because you're going to notice that a lot of the properties of glycosaminoglycans actually stem from this, the fact that they're very negatively charged molecules. And then the rest of this just talks about the structure of it, but we're going to go over it. But one of the key concepts, again, is that it's a repeating disaccharide unit. Disaccharide means two sugars, and it just means that it has a pattern. It's going to go A, B, A, B, A, B. And again, we're going to look at what that actually looks like later on. Okay, so remember how I told you that glycosaminoglycans are repeating disaccharide units? Let's look at what a disaccharide actually is. Right over here in the middle, I have my disaccharide, which again means two sugars. So I have one sugar on the left and I have one sugar on the right. And what's cool about glycosaminoglycans is that one sugar is going to be what I call my acidic sugar, and the other sugar is going to be an N-acetylated amino sugar. So something like N-acetylated glucosamine, for example, or galactosamine. So one is going to be an acidic sugar, and we're going to go over. And on the left over here, you can see examples of acidic sugars. So D-chloric acid is an acidic sugar. L-hyaluronic acid is another acidic sugar. So it's going to be one component of it. And then the second component of it, like I said, is going to be N-acetylated amino sugar, so something like N-glucosamine and galactosamine, which you can see on the right side over here. The other thing that I told you is that it's really important to know that GAGs are highly negative. And so we're going to talk about three components that lead them to be highly negative. And I think it's really important to know. One of them is a carboxyl group, which you can see over here on my acidic group. I have this blue, and this is my carboxyl group. I know it doesn't look negative right now, but you have to understand that in our body, this is actually going to be depronated. Depronated means that this proton over here, this hydrogen, which is also called a proton, is going to be lost. And because hydrogen is positively charged, and because I'm losing a positive ion, it's actually going to make the rest of the molecule negative. So again, depronation is removing positivity, which in other words is increasing negative charge. And so one of those things is a carboxyl group. The next thing that we can look at over here is this amine group. And it's a very similar concept where if you look at the amine group over here, I have 2 hydrogens, but the amine group over here only has 1 hydrogen because I've replaced the second hydrogen with this group over here, which is called an acetyl group. The thing is, an acetyl group is not negative or positive, it's just neutral. So in this case, I'm losing a positive ion and I'm replacing it with something neutral. So I'm not gaining negativity like I was with my carboxyl group. Instead, I'm just losing positivity and replacing it with something neutral. But the overall effect is still going to be increasing negativity, right? And then the last one they don't show over here, but in some sugars, they're going to add a sulfated group. And you can notice that the sulfated group has a negative charge. And so again, I'm increasing negativity. So I wish they had drawn this in the proper way. It should be COO minus, and that minus is negative. So that should give you an indication that it's increasing negative charge. I'm also decreasing positive charge by removing one of the hydrogens of the amine group and replacing it with something neutral. And then lastly, and I know they haven't shown it here, is that on one of these sugars, you can also add a sulfate group, and the sulfate group is also negative, which is also going to increase the negative charge. So I would be sure I know about all these 3 components of GAGs. Okay, so why is this important? Why do I care about the fact that GAGs are negatively charged? The cool thing is that if I look at a joint over here, okay, inside of my joint, I have like cartilage and lubrication fluid. And the thing about my joint is that as I'm walking, for example, like when I'm walking, my knee joint is going to get compressed, which is what they're showing over here. Like this black circle is the joint. And then inside all these red lines are the glycosaminoglycans. And then this sort of like yellowish background is the water. And so they're showing you it normally over here. And then they're telling you that once you compress a joint, all the water is going to escape, but the glycosaminoglycans are going to stay. Here is why it's so important that glycosaminoglycans need to be negatively charged. It's because water is polar, right? And that the 2 hydrogens of water are positive or they're slightly positive. And so they are going to be attracted to the negativity of the glycosaminoglycans. In other words, the glycosaminoglycans, after the joint has been compressed, the glycosaminoglycans are actually going to attract the water back into the joint. Because again, the glycosaminoglycans are negative and the water is positive and it has a slight positive charge. And so it's going to be attracted back into the joint. And this is really important because imagine if the glycosaminoglycans weren't negative. That means that every single time I went walking and every single time I compressed my joint, the water would never be drawn back in, right? And so that's why we call glycosaminoglycans lubricants, is because they attract water or they attract fluid back into the joint. And by attracting fluid or water back into the joint, it's going to reduce friction. Friction is when my 2 bones rub against each other. And obviously I don't want that. So the whole point of glycosaminoglycans is that it acts as a lubricant. And the reason for that is that it stems from the negative charge of the glycosaminoglycans.
It absorbs the shock?
That's one of the other things. Yeah, shock is kind of different. Shock is when I have my glycosaminoglycans and I have my water. The water is technically what absorbs shock.
Okay, thank you.
Okay, so over here, I just want to introduce the concept to you of aggregants. I'm going to describe it later to you, but what I want you to know is that aggregants are made up of hyaluronic acid as well as some other components, but I want you to at least remember that aggregants are made up of hyaluronic acid, and that the hyaluronic acid is non-covalently attached to proteoglycans. So again, an aggregant, I'm going to show you in an image, so if you can't picture it right now, that's okay. But I just want you to be exposed to the idea that an aggregant is made up of a hyaluronic acid that is non-covalently attached to proteoglycans. Okay, just at least be exposed to that concept of what an aggregant is. I'm going to show you in an image later. And then they just talk about some other glycosaminoglycans that we're going to discuss right now. The other thing they want you to know about hyaluronic acid is that it's very, very, very large. And the cool thing about that is that because it's so large, it's going to displace a large volume of water. which if I show that in the previous slide, you can imagine if I'm displacing a large amount of water, it also means that when the joint relaxes, I'm also attracting a large amount of water. Does that make sense? So because hyaluronic acid is so giant, it means that it's going to displace amount of water, which means a lot of water might leave when my joints compress. But at the same time, it means a lot of water will also enter when my joint is relaxed. So the main thing is that because hyaluronic acid is such a giant molecule, it's going to attract a lot of water. And because it's attracting a lot of water, it makes a very good lubricant. That's sort of the main idea. Okay, so over here, we're going to specify hyaluronic acid. Over here on the right side, you can see that they're comparing the size of hyaluronic acid to other things, and you can see it's much, much, much more giant compared to some of the other ones. Like look at this globular protein and then look at hyaluronic acid. It's a very large molecule. Okay, so remember earlier on, I told you that hyaluronic acid is going to make up a structure called an Agrican. And I told you something very specific about the bond. Do you remember what I told you about that bond? What was a property of that bond? The bond between the hyaluronic acid.
Non-covalently attached.
Exactly, it's non-covalently attached. Non-covalently attached. I just want that to be really clear. We're going to go over the structure of an Agrican later. The other thing is that I just want you to know that obviously hyaluronic acid is in articular cartilage. Articular cartilage is also where I'm going to find the aggregants that I keep referring to. Okay, so aggregants are in high amounts in the articular cartilage. And so it should make sense to you because I told you that aggregants are made up of hyaluronic acid, that you're also going to be finding hyaluronic acid in the cartilage. All those ideas should connect to you, hopefully. The other thing that I'm going to be showing you on the next slide is the synthesis of hyaluronic acid because it's a little bit unique. So usually I think you guys know that the way that something is formed is that it goes maybe like to the Golgi to the ER, sorry, and then like to the Golgi, or sorry, the ER and then to the Golgi and then it leaves, right? That's like normally how like proteins and stuff are formed. Hyaluronic acid is different. Instead of being formed inside of the cell, it's actually formed on the cell membrane. This is very unique. I don't think we've ever heard of something that's been formed on the cell membrane. And if you think about this logically, it should make sense. One of the points that I've really emphasized about hyaluronic acid is that it's huge, is that it's giant. And so it doesn't make sense that if I have such a giant molecule to make it inside of the cell. Because it's so large, I actually prefer to be making it within the extracellular matrix or actually outside the cell. So you can notice I have like this protein over here, and this protein is synthesizing this hyaluronic acid, and that the hyaluronic acid is being synthesized outside of the cell. Or the other way you might hear it is that it's being synthesized on the cell membrane, because technically the protein is on the cell membrane. That's a more accurate way to say it. You should say it's synthesized on the cell membrane, okay? And so pretty much you can see that I have all the precursors of hyaluronic acid. I'll show you them in precursor. You see the structure of hyaluronic acid on this one. I have my acid that I told you about, and I have my N-acetyl sugar, right? We talked about how there's a disaccharide, one has to be an acid, one has to be an N-acetyl sugar. So they've just shown you that those two components are just floating around in the cytosol, and then they're going to go to the protein that's on the cell membrane. This is a very unique feature of hyaluronic acid. The protein that synthesizes hyaluronic acid is on the cell membrane, and the hyaluronic acid is going to be formed in the extracellular matrix. So that's another thing that they want you to know. So this last point over here is that the sugars are in the cytoplasm, and that they go to a protein that's on the cell membrane, and then the hyaluronic acid is going to go directly into the extracellular matrix. Okay, so now I'm going to quiz you. So the precursor sugars, these sugars that make up the hyaluronic acid, where were they? Were they inside the cell, outside the cell?
Inside the cell.
In the cell. Exactly, yeah. And then where was the protein that synthesizes hyaluronic acid?
In
the cell membrane.
Exactly. So the sugars are in the cytoplasm, and then they're going to go to the protein on the cell membrane. Perfect.
Can I ask you a question?
Mm-hmm.
Since we have a sugar and then we have an acid, and acids, we usually know that acids are so positive, but we need to make the gags very negative. Does this mean that the sugar basically makes it more basic, or it has nothing to do with that?
It's more so that this acid, because it's an acid, can react with other basic or other alkaline compounds in the extracellular matrix. But these two are not, if this was a base that was neutralizing this, then this wouldn't be an acid anymore. Then this wouldn't
Yeah.
have a negative charge. They're not bound by the acid component, right? They're bound by this other bond over here, which is called a glycosidic bond. They're not
Glycosyl.
technically like each other. Not the acidic part of it. Does that make sense?
Yeah. But like the gags in general are negatively charged. So that's what confused me a little bit. Because we know that acids are more like they're mostly positive, and then the gags are negative. So that's what confused me.
Oh, okay. I don't usually think of acids as generally being positive. Although I guess you're saying that because of the H plus, right? Because of the proton. Yeah, I guess I could see that. Yeah. So I guess in this way, I don't know, we call this part the acid, like the COO.
I'll stick to what's in the slides, to be honest.
Right?
I'll just stick to what's on the slide. I don't need to make think pieces, to be honest.
Okay. And then we can go over, so that was one glycosaminoglycan, was hyaluronic acid. And then, so there's these other ones that I briefly mentioned before, but we have chondroitin sulfate, dermatan sulfate, and keratin sulfate. I'm going to go into more depth about these later, but I just want you to notice that chondroitin sulfate and keratin sulfate are both also found in the cartilage. So far what you know about cartilage is that you know you have the structure called an agrican, and you know that within the agrican, I have hyaluronic acid, and that the hyaluronic acid is non-covalently attached to proteoglycans. What I haven't told you is that the proteoglycans are actually made up of chondroitin sulfate and keratin sulfate. Again, I'm going to show you what that looks like in an image later, but I just want you to know that all of these ideas connect. I have aggregants. Aggregants are made of hyaluronic acid plus proteoglycans, and proteoglycans are made up of chondroitin sulfate and keratin sulfate. Okay, which means that you're obviously going to be finding chondroitin sulfate and keratin sulfate also in the cartilage, because aggregants are found in the cartilage. And then they are just showing you the structures over here. I do want you to pay attention to these sulfurs. You don't have to memorize these structures, obviously, but I just want you to be familiar with these sulfurs, because when we go over later the enzyme deficiencies, I think if you're at least familiar with the structures, you'll be able to recognize which sugars are being affected based off of where the sulfurs are. So for example, I just want you to appreciate that chondroitin sulfate has a sulfur group, and that you can see keratin sulfate actually has 2 sulfur groups as well. The other thing I really want you to notice about keratin sulfate is that, remember how I told you that one of the components of the disaccharide is almost always a sugar. I'm sorry, it's almost always an acid, right? Uramic acid in this case, like glycouramic acid. Keratin sulfate is an exception to that. Instead of this being an acid, I instead have galactose. Okay, so keratin sulfate is not made up of an acid, it's made of galactose, okay, plus the N-acetylglucosamine. But the main thing is that it's special in the way that instead of an acid, it's replaced with galactose. I think this is just important for like MCQs questions or something like that, because usually when something is special, they like to test on that.
Can you repeat?
Okay, so just remember that earlier on in this presentation, I told you that that gags are always made up of an acidic sugar plus an N-acetylated amino sugar, right? I told you that that's always a component of the gags. Keratin sulfate is a specific gag that is an exception to this, because it does not have an acidic sugar. Instead of an acidic sugar, it has galactose. Okay, so if you look over here, you can see instead of an acidic sugar, I have galactose in keratin sulfate. Is that clear?
Yes, thank you.
Okay. Okay, so now we're going to move on to two new GAGs. One is called heparan sulfate, and the other one is called heparin. Heparin, I don't know if you guys have heard of, I know it's early for year one, but this is a really common anticoagulant that we use as medication in clinical practice. But the main thing I want to focus on when you're looking at these two GAGs, obviously you don't have to memorize the structure, but I want you guys to look at them and look out for the sulfur groups, okay? Just try to recognize sulfur groups. So SO3 minus, SO3 minus, SO3 minus. You guys tell me, between heparan sulfate and heparin, which is more sulfated? Which has more sulfur groups? Look at the structures. How many sulfurs? Perfect. Yeah, heparin. So the reason I'm bringing this up is because they specify it in the next slide. And I feel like it sounds kind of counterintuitive, right? If I just told you the name, if I just told you heparin sulfate, and I told you heparin, you might think, oh, heparin sulfate has sulfate in its name, so it must be more sulfated. That's not true. I don't know why they named it like this. I know it's really confusing, but in actuality, heparin sulfate only has one sulfate group, but heparin has 3 sulfate groups. So heparin is actually more sulfated compared to heparan sulfate. The other way, sorry?
I have a question. Will they ask us to identify the location of the sulfate?
I don't think so, but I think they might ask you which is more sulfated, because that is written on the slides or something like that. So I only drew it out so that you guys can see what I'm talking about. Because again, I feel like if I said it out loud, heparin sulfate is less sulfated than heparin, I feel like that was confusing. So you definitely don't need to memorize these structures. I don't know how much they want you to identify, but I think at the very least you should know that heparin sulfate is less sulfated. Is that fair? Is that okay?
Yes, yes.
Okay. The other way that you might hear this phrase is that you might hear that heparan sulfate is more acetylated, COCH3, this is the acetyl group, than heparin. Look at heparin, I don't have a single acetyl group, but I do have an acetyl group in heparan sulfate, which is this thing over here. So again, another thing that you might hear is that heparan sulfate is more acetylated compared to heparin. I'm going to test you on this on the next slide. Okay, so for heparan sulfate, does it have more, I forgot which one this was. Does it have more sulfate groups or less sulfate groups compared to heparin?
Less.
Less, right? And does it have more acetyl groups or less acetyl groups?
More acetyl groups.
More.
Perfect. Yeah, so it's higher acetylated, but heparin is more sulfated. Perfect. And then I just kind of briefly talked about that heparin is an anticoagulant. It's actually a very common medication that we use. And it's also actually found in mast cells. If you guys know mast cells, that's responsible for example, like allergic reactions. So the main idea here is that it plays a role in the immune system and like allergic reactions and that kind of stuff. I don't know how much you guys know about mast cells, but pretty much what happens is that if you have like an allergic reaction, for example, if somebody eats peanuts, what happens is that mast cells get attracted to whatever area the peanuts were at, and they're going to release a bunch of granules. And the main granule is actually histamine, but some of the granules are also heparin. I don't know if you need to know that level of detail, but I would at least know that heparin is found in mast cells, which mast cells are a component of our immune system. They play a large component in allergies, and that it's also a common anticoagulant medication. Okay, so this, what you're looking at right now is you're looking at a proteoglycan. If you attended my talk yesterday, you know the thing that I always say to myself is proteoglycan is a glycan, which means that proteoglycan is a sugar. And what that means is that it's mostly carb and just a little bit of a protein, just a little bit of a protein core. Okay, so proteoglycan is mostly carb and it just has a little bit of a protein. Okay, so that's what they're showing you over here. They're showing you proteoglycan. They're showing you the carb component of it, which is what I'm highlighting over here. And they're showing you the protein part. And what they want you to be aware of is they want you to be aware of how the protein connects to the carb component or connects, to the sugars. Okay, and that so if I look at my carbs, this sequence is really important. So look at the sequence of each sugar. It goes glucuronic acid, galactose, galactose, xylose. And you'll notice that the xylose is attaching to the protein. Okay, so my xylose is a sugar and is the specific sugar that attaches to my protein. That is really important to know. And that what we call the tetrasaccharide linker is pretty much that the 3 saccharides that are going to be behind the xylose is always going to be in this order. It's always going to be glucuronic acid, galactose, galactose, xylose. And when we talk about how proteoglycans are made, you're going to see why this order always exists, but it's just important to know that. I don't know if you need to know the exact one, but I'm going to be going it over so many times that I think you'll memorize it by the end of the lecture anyways. But you should know that this specific sequence always exists right before the attachment with the protein, and that specifically the xylose sugar is what attaches to the protein. On the flip side, the part of my protein that always attaches to my sugar is serine. So this part over here is serine. Serine is an amino acid. If you remember, amino acids are what make up proteins. And the specific amino acid that is always going to be attaching to my sugar is the serine amino acid. I really think you need to know that. I think you need to know that xylose is the sugar that always attaches to the protein, and that serine is the amino acid that always attaches to the sugar. And then just so you guys are aware, I told you that this.
So I have a question.
Mm-hmm.
Xylose attached to the protein and serine attached to the sugar, right?
Yeah, they're attached to each other. Look, this is xylose and this is serine. They're attached to each other. But this serine is an amino acid, so it's considered the protein part of it, and xylose is a sugar, so it's considered the carbohydrate part of it.
We have to memorize the structure, because I don't remember we have to memorize the serine structure. Or
No. The structure, definitely not. No, but I do think that you should know that serine is the amino acid that connects.
All right. Is it always the case that the serine is the amino acid that connects the protein to the sugar, or like we have other cases?
No, I think it's only serine.
So
Only
serine
serine and only xylose, yeah.
So xylose is XYL, which is connected to serine.
Exactly. And
Okay.
xylose is a sugar and serine is an amino acid. So can you guys see that? Can you guys see the connection between the xylose and the serine? This is serine. I know it's not labeled, but you can see the serine is part of the protein, but my xylose is part of my carb chain. That's the main thing that I wanted to emphasize on. The other thing that you guys are going to notice is that this tetrasaccharide linker, remember how I told you that glycosaminoglycans are always made of disaccharides, like this repeating unit over here? I'm going to show you it later, but just so you guys know, the disaccharide unit is just going to add on to this tetrasaccharide linker. It will make more sense when I talk about the synthesis of it, but just so that you guys know, like these disaccharides are going to attach over here to this glucuronic acid, and they're just going to keep expanding. Okay, so now I'm going to quiz you guys. So I told you that the proteoglycans are mostly sugars, but they also have another component to it. Do you guys remember what the other component to it is? There's a sugar and there's a
Protein.
Exactly, specifically a protein core. over here, or a core protein. But in proteoglycans, which is more? Is the carbohydrate more or is the protein more?
Carbohydrate is more.
Perfect. Yeah, the carbohydrate is going to be more. Okay. And then we already went over this sequence over here: glucuronic acid, galactose, galactose, xylose. Okay. And I told you that the xylose is going to attach to serine. Serine is an amino acid. Oh, I just spoiled the next part. Okay, so you guys tell me. So which sugar is the part that attaches to the protein? What's the name of the sugar that attaches to the protein?
Xylose.
Xylose.
That's perfect. And what is the name of the amino acid that attaches
Serine.
to the sugar?
Serine.
Perfect. Yeah, so you guys got it right. Okay, so now remember how I kept hinting to you guys that we're going to talk about aggregans. We're going to talk about aggregans. We're finally on the slide about aggregans. Okay, so aggregans, I really want to make sure you guys know aggregans are found in cartilage. Okay, so everything that you're looking about, just imagine that it's in cartilage. And then it acts as a lubricant, that kind of stuff. So this entire structure over here is an aggregan. Okay, and this bold black line down the middle, this is my hyaluronic acid. Remember the GAG that I told you about that's really, really huge and that displaces a lot of water and is like made on the cell membrane? That same GAG that I was talking about earlier, that's what I'm pointing at over here. It's called hyaluronic acid, okay? And you can see that the hyaluronic acid is non-covalently attached to these structures over here, okay? And I call these structures peptidoglycans. Peptidoglycans you already know about. You were just looking at a peptidoglycan. This is a peptidoglycan or a proteoglycan. It's the same thing, right? So this is the protein core that we talked about in the last slide, okay? And then these little segments that are sticking off like leaves, these are the saccharides or the oligosaccharides or the carb component. Okay, so I'm going to say this entire thing one more time. This is an aggregan. This entire thing is called an aggregan, okay? Then my aggregan, I have this hyaluronic acid, this bold line down here, and my hyaluronic acid is non-covalently attached to this structure over here, which is called a proteoglycan. And the proteoglycan is made of a protein core, which I can see in the middle, and is also made of saccharide chains sticking out over here like leaves, okay? Specifically, what you need to know, not saccharide chains, they're attached to other GAGs, specifically chondroitin sulfate and keratin sulfate. Remember earlier, I told you that peptidoglycans are made up of chondroitin sulfate and keratin sulfate, right? So this is my proteoglycan over here. So this and this are the same thing. Okay, this structure that you're looking at over here on this slide and this structure that you're looking over here on this side are the same thing. Okay, this black line in the middle is my protein core. And then these things that stick off like leaves are GAGs. I said sugars before, I was wrong. I'm so sorry. They're GAGs, they're glycosaminoglycans, which technically is a sugar, but it's a modified sugar, right? The highly negative sugar. And I want you to know specifically that they're GAGs. So over here,
Sorry,
these
could
things.
you repeat it again? But I
Okay.
confused a little bit.
Okay, sorry. So this is my aggregan. Everyone's
I
following
have
that.
a question.
Mhm.
Aren't proteoglycans the same as glycosaminoglycans?
Proteoglycans are
Or
made
proteogly-
up of glycosaminoglycans. So this is a glycosaminoglycan. And then if you add my glycosaminoglycans plus my protein, now I call this entire thing my proteoglycan.
But aren't proteoglycans made of glycoproteins?
Proteoglycans are made of lipoproteins?
Yes.
I don't think so. I've never heard of that. I don't know. See, basic proteoglycan is made of a core protein, and it's also made up of GAGs. So the core protein is attached to the GAGs. Does that make sense? And lipoproteins are something else. Those are lipid proteins. It's an entirely different structure. So I'll just explain it one more time because I know I messed it up. But this entire thing is my proteoglycan. This thing down the middle is the protein. And then these things that are sticking off like leaves are my GAGs, my glycosaminoglycans. Remember, we've been talking about glycosaminoglycans this entire time. All the other slides that I've talked about, heparin sulfate is a glycosaminoglycan. Heparin is glycosaminoglycan. Chondroitin sulfate, dermatan sulfate, keratin sulfate, all of these are examples of glycosaminoglycans. And I told you that glycosaminoglycans are disaccharides, so they are sugars, but they're sugars that have been specifically modified to be very highly negative. Okay, so just imagine these glycosaminoglycans. Imagine these as those little leaves sticking out over here. And the glycosaminoglycans are being attached to a protein core that runs down the middle over here. And now I call this entire structure my peptidoglycan. And my peptidoglycan is non-covalently attached to my hyaluronic acid. And when that happens, I call this entire structure my agrican. Is that more clear this time?
The
Sorry,
hyaluronic
but here,
does it has sulfur
Hyaluronic acid.
or like it's not sulfated, right?
Yeah, you're right.
the-
You're right. There's no sulfur on hyaluronic acid.
Okay, and what I seriously don't understand, like what's the function of the heparin? I
What's
know
the,
that, yeah, the heparin here, these.
the heparin?
Yeah, heparin. Sorry, I mispronounced it.
No, you're good. So you mean like what does it do naturally in our body?
Yeah.
So naturally in our body, all you need to know is that so there's these cells, okay? And the cells are called mast cells, okay? And the mast cells are part of our immune system. And specifically when mast cells are activated, they're going to induce, right?
Yeah. Yeah.
Right? So inside of my mast cells, if you imagine a mast cell, let me draw it. So imagine a mast cell over here. Okay, this is an immune cell, and there's going to be a bunch of stuff within it. One of the things that's inside of a mast cell is heparin.
So heparin is a gag, or heparin is it's a component of the gags.
No, heparin is a gag. Heparin is a gag.
All right, so we're just specifying the type of gags. Okay, thank you.
Exactly. These are all different types of gags. Heparin sulfate, chondroitin sulfate, dermatan sulfate, all of these are different types of gags. Hyaluronic acid is a different type of gag as well.
Yeah, the hyaluronic acid basically makes up the aggregants.
Perfect, yeah. Is this clear what an aggregan is?
Yeah, it's made up of proteoglycans, which means we have a protein core, which is the protein core is being connected by the xylophane or like the XYL sugar, and through an amino group called the I'm not sure, I forgot, but it's
Serine, yeah.
serine, yes, serine, and then the proteoglycans are being connected by the hyaluronic acids.
And what's the bond between the peptidoglycan and the hyaluronic?
Non-covalent.
Perfect. Yeah. Okay, so like I talked about before, we're looking at the same thing. We just zoomed in. Okay, so imagine that I zoomed in on this section over here, for example. That's what's going to be on the next slide. So I still have my hyaluronic acid as the core, or whatever. The hyaluronic acid is over here, and it's non-covalently being attached to this structure over
sorry, I
here.
have a question here.
Sorry?
Is the hyaluronic acid not a part of Agrican because they circulate in just only the protein core and the glycosaminoglycan?
Are you asking if hyaluronic acid is part of the Agrican?
Yes. Is it part of the Agrican? Because they circle here It's not consisting of hyaluronic acid.
Over here, okay, are you talking about this part over here?
I think what he means is that in this image, there's a circle in the Proteoglycans, and it doesn't connect, like it doesn't circle the whole thing. So in the circle itself, it's only circling an Proteoglycans, and it's saying this is an Agrican. That's what his question.
Okay.
So he's saying is the hyaluronic acid part of the Glycans?
Okay, that's a good question. From what I could look up and what I looked into, I think it is considered part of the Agrican. I guess you can consider like this segment, for example, of the hyaluronic acid is still being included in the entire Agrican. But that is a good question. I understand what you're saying. But does that make sense? Like it still is kind of part of it. It's just that we localize sort of one specific aspect of the hyaluronic acid. I still would consider hyaluronic acid to be part of the Agrican from what I've looked into. Does that make sense?
I mean, it makes sense to me. It's
Clear, thank you.
Okay.
Mhm.
So the other thing that they want you to know about is that I have my peptidoglycan. So again, I'm looking at my protein core. And then I have my gags that I talked about, right? That attach onto my protein core. So what they want you to know, I think, is that the ones at the top are usually chondroitin sulfate. So the gags that are at the top are usually chondroitin sulfate. And the gags that are at the bottom are usually keratin sulfate. Okay. And that the other thing that they want you to know about is that the way that the proteoglycan attaches to my hyaluronic acid is using this link protein. Okay, so I need this link protein in order for my peptidoglycan to attach to my hyaluronic acid. Okay, and the reason that's
covalent bond, and we have two bonds, the link protein and the non-covalent bond, right?
I think the link protein attaches to the hyaluronic acid using non-covalent bonds.
Yes.
Okay. So sorry, I lost my train of thought. So the reason that they want you to know about that is if I don't have my link proteins, I'm not going to be able to connect my proteoglycan onto my hyaluronic acid. And so I'm not going to be able to form the aggregan properly. And the problem is that I told you that aggregants are made, are found mostly in a specific kind of tissue. Does anybody remember where I told you that aggregants are mostly found?
Articular cartilage.
Perfect. They're mostly found in cartilage tissue. So what that means is that if I don't have a link protein, I can't make my aggregan properly. And if I can't make my aggregan properly, I'm going to have malformation in my cartilage. Okay, so now, yeah.
Can you repeat just like what happens if there is no aggregans?
Okay, it's not that you don't have, well, I guess so, yeah. Like, if you don't have a link protein, you can't form the aggregants properly. And
what's the result? Like, what happens?
It's just the deformation of the cartilage. And so, for example, like delayed bone growth, if you remember when kids are very young, the reason that their bones are growing is that actually the cartilage is growing and then it's being ossified to bone. And so you can imagine if it's happening in a young kid, they're not going to be able to grow their bones properly. The main idea is that it leads to defective cartilage formation. Another example of that is that they specify like craniofacial abnormalities. If you know your nose, the nose bridge is actually partially made out of cartilage. So again, if I can't form my cartilage properly, I'm not going to be able to form my nose properly. And so I'm going to have facial abnormalities. That's another application of this. Does that make sense?
Yes.
And then just so you guys know that the link protein is also called adherin. And so that's all they're telling you over here. They're saying that if you don't have the link protein, you cannot anchor the proteoglycan onto the hyaluronic acid. That's the main concept that they're trying to convey. So an aggregant is a proteoglycan with the linker proteins, yes. I believe the linker protein is considered, you can see it's being circled. So linker
So
protein.
I have a question.
Mhm.
In Agrican is consists of chondroitin sulfate and keratin sulfate.
Yes, yeah.
Okay.
And then you can see where they are, right? They're specifically.
Yes. Yeah. Okay. Thank you.
Okay, so now remember earlier on, I told you that we're going to talk about how we make GAGs. So the specific example that they have for you is chondroitin sulfate. So I know it looks kind of intimidating, but let's just break it down. So over here, I have my protein, right? Okay, somebody's asking for 10 minutes to pray. Yeah, we can take a 10-minute break to pray. So you want to be back by 12? Okay, we can resume by 12. So we're going to be continuing the part where we're actually going to be synthesizing a glycosaminoglycan. So I've actually referred to this earlier on. So now I'm going to be showing you how do we actually make a glycosaminoglycan. And the specific example that they've shown you over here is chondroitin sulfate. Okay, I just want to set the image. I want to see what we're talking about over here. So this green part over here is our protein core. Remember how I told you there was a protein core and that you have to add glycosaminoglycans to it? So now I have my protein core and you want to add a glycosaminoglycan. And I told you that the protein core, I covered it now, but the protein core has serine, which is the specific amino acid that is going to attach to the sugar component. Do you guys remember the name of the monosaccharide or the name of the sugar that attaches to serine?
XYL.
Exactly, which is like shorthand form for xylose. Perfect. Okay, so that's what you can see going on over here. The green that I've covered up, let me just erase it. The green is the protein core that we talked about before, remember the protein core that we were talking about before? So now we're imagining this protein core and we're talking about how do we add on the glycosaminoglycan, right? So this is my protein core. The same thing as over here. And then I'm adding on a glycosaminoglycan, which is this thing that's being added on over here. And we already know that the serine of the protein must attach to the xylose of the sugar segment. And if I told you there was a very specific order, right? So it has to go xylose, and then I have to add 2 galactose, and then finally I have to add glucuronic acid. That specific sequence has to happen. So after I add in the xylose, I have to add 2 galactose, and then I have to finish it off with the glucuronic acid. That's the tetrasaccharide linker that we talked about afterwards. Once I have my linker, once I have this final 4 sugars complete, now I can start adding my disaccharide units. Okay, and I can add as many as I want. Remember before when we talked about disaccharides over here, I told you one has to be an acid, one has to be a sugar, like that kind of stuff. So we can add on these disaccharide units. We can add them onto my linker. The main idea is I need my 4 glucuronic chain first, okay? So that's sort of a revision of what we've covered before. Hopefully now it's a little bit easier to visualize. But the other thing that they want you to know about is that I can't just add on my sugar directly. Okay, you see this sugar, I can't add it on directly. I first have to add on this molecule, which is called UDP, to my sugar. And then once my UDP is attached to my sugar, now I can incorporate it into the chain. Okay, so if you notice, all of the sugars that are being attached to the chain are always UDP plus the sugar, UDP plus the sugar. You do eventually lose the sugar, you do eventually lose the UDP. The UDP doesn't stay inside of the actual chain once it formed, but you need UDP attached to my sugar in order to attach the sugar onto the saccharide chain. Okay, that's one of the new concepts that they're trying to explain to you. You can't just attach a sugar directly. You need to add UDP first. Okay, which they've shown you over here. They've shown you, for example, a sugar plus the UTP.
I'm sorry. I joined really late, so I couldn't catch what you said. Can you repeat it?
Okay, do you want me to explain the UDP part, or do you want me to explain the serine and the sugars being added as well?
Like, I want you to repeat everything after the break, if you don't mind.
Okay. I'll also quiz people at the same time. So for those of you that were here, it'll also be beneficial to you because I'll also be quizzing you. Okay, so pretty much what I explained is that this green part over here is my protein core. If you remember my protein core, remember the protein core, right? And we said that the protein core is attached to glycosaminoglycans, right? This part over here. So now we're just showing you how does it get added? How do I have a protein core and how do I add glycosaminoglycans to that? So they're just showing you, over here's the protein core and they're adding in glycosaminoglycans. Okay. So, and then remember the revision from last part, I told you that for my protein core, the specific amino acid that needs to attach to the sugar is the serine amino acid. And then I want you to tell me what was the sugar that needs to attach to the serine.
The XYL.
Exactly, xylose. Okay, once I add in the xylose, you need to add 2 more specific sugars. It has to be this sugar. What is the name of this sugar that I need to attach to my system?
The gallic acid.
Lactose.
The lactose, exactly. After I add my xylose, I need to add 2 galactose. And then what's the last thing that I add in my tetracycline chain?
Hyaluronic acid.
Glucuronic acid, perfect. So now you know your tetracycline chain by heart. You literally already know it. You know the entire sequence, which is perfect. So xylose, galactose, that's why these 2 look the same. And then it ends with glucuronic acid. Someone's asking, why do we have 2 serines on the core protein? It's because if I wanted to, I could add a saccharide component over here as well, right? Look at my core protein. I have multiple points that I can add saccharides. So over here is a serine, over here is a serine, over here is a serine. Does that make sense? Does that answer your question? Why I have more than 1 serines on the core protein? Because if I wanted to, I could also add a saccharide here. Okay. And then the other thing that I was explaining is that the new concept that's being introduced over here is that I have my sugar. In order to actually attach my sugar to my core protein, my sugar first needs to attach to a molecule called UDP. Okay, so first the sugar plus the UTP need to bond. And then after I have the UDP attached to my sugar, then I can incorporate the sugar onto the saccharide chain. Okay, so attach the sugar directly. It needs to be attached to the
The
UDP.
UTP energy resource, the same as ATP.
Yeah, you can think of it like that.
Okay, so basically we need energy to connect the sugars and the protein.
Exactly. And the specific form of energy is UDP.
Yeah. And we
And the
phosphorate it or like dephosphorate it. So we use 1 phosphate group and we add them together, right?
You release 1 phosphate group. And then to be honest, I don't know the details of the exact reaction, but I just know that you add the UDP and that it's necessary for the attachment of the sugar, but the UDP does not stay, like it doesn't stay attached to the sugar. It's only needed temporarily for the attachment.
So that we can activate it and connect them.
Yeah, exactly. Okay, those are the main concepts. And I think they just want you to know one more thing. The enzyme that's responsible for actually attaching the sugar to the saccharide chain is called Glycosyltransferasis. I think they want you to know that.
It attaches what to what?
The Glycosyltransferase, it attaches, you remember the whole process that we're talking about? We're slowly adding sugars onto the saccharide chain.
Yes.
Right? So the enzyme that attaches this sugar over here onto this chain is called the Glycosyltransferase. So it attaches the sugar to another sugar, for example, like this diamond is being attached to this circle through the enzyme glycosyl.
This is the disaccharide units, right?
This actually is not the disaccharide unit. Oh, it's forming the disaccharide unit. Yeah. Once
Okay.
these two form together, this is a disaccharide unit right over here.
Thank you.
Okay, so now we're going to be looking at it, but we're going to be looking at it from the perspective of a cell. And a lot of this is actually revision. Okay, so you guys already know how proteins are made, right? They're made first from the ER, and they need to be translated. So that's what they're showing you over here. And then we talked about how we need to add xylose, right? Which is a sugar, to the serine of the protein, right? What were the two sugars that I then add to xylose? What are the two sugars?
Galactose and galactose.
Exactly. And then what's the fourth one? What ends my tetracycline chain?
Glucuronic acid.
Glucuronic acid. Perfect. You have your tetracycline.
Never mind.
And then once you have the tetracyclic chain, now I can start adding as many disaccharide units as I want. I can just keep expanding the disaccharide units. The main concept is you need this tetracyclic chain, which you guys already know by heart already, which is amazing. Okay. And then once I make, once I have this protein core, which is this orange thing in the middle, then I have these little brown things sticking off, which are my gags. I now have my peptidoglycan, and I can now release my peptidoglycan into the extracellular matrix. Okay. The peptidoglycan is going to non-covalently attach to what is it? What's the name of that gag?
Hyaluronic acid.
Yes, both are correct, hyaluronic acid and hyaluron. So the peptidoglycans, and I believe what they're trying to show you is that these are the proteins. Remember in the previous slide, there's the G2 and the G1 and the G3. I believe that's what they're trying to show you over here, that you also can have these proteins. And that you need these proteins to attach my peptidoglycan to my hyaluronic acid. And where was hyaluronic acid, the protein that synthesizes hyaluronic acid, where is it found?
On the plasma membrane.
Cell membrane.
Yes. So you guys got everything about this.
I have a question. What do you mean by peptidoglycan?
So peptidoglycan is this entire structure over here. It's the protein plus the gags sticking out of it.
Okay, thank you.
Okay, so now we are going to talk about heparin, or two functions. Sorry, not a heparin sulfate. We're going to talk about two functions of it. Let me make sure I'm not saying that wrong. Yeah, we're going to talk about two functions of heparin sulfate. So the first function of heparin sulfate is over here. If you remembered, I mentioned that heparin is an anticoagulant, and I guess they also want you to know that heparan sulfate can also be one, although the one that it mentions on your slide is heparin. So in an MCQ, if they asked me what an anticoagulant was, I would go with heparin, not heparin sulfate, but the image that the doctor did put, like in your slides, it does show heparan sulfate as well. But I still want you to know, when you think anticoagulant, please still think heparin as your first one. It's just that heparan sulfate can also act at it. Like, it would be my second choice. If we have heparin, go for heparin for an anticoagulant. But the doctor did put pictures of heparin sulfate being an anticoagulant. So that's why I'm also showing you that. Okay, so now we're going to talk about, so I have my heparin sulfate, which is over here, right? It's my heparin sulfate. These green boxes are areas of my heparan sulfate that are very, very sulfated. They're very high in sulfur, okay? And we call these my NS domain. Okay, so this green box over here is a segment of my heparan sulfate, and it's very high in sulfur, and we call it my NS domain. Before I go into this, actually, I just want to give a background of how clotting works, because I don't know how much you guys know. Pretty much we have these things called coagulation factors, okay? There's like factor 1, 2, 3, 4, blah, blah, blah. And one of those factors is factor 10A. Okay, if factor 10A is present, what you need to know is that coagulation will occur. Okay, factor 10A is important for coagulation. Okay, or it's a procoagulant or whatever. So what you need to know is I have this thing called antithrombin. Coagulation and thrombin are kind of like two sides of the same coin. Coagulation is when it happens normally. Thrombosis is when I have abnormally high levels of coagulation. Okay, so you can imagine that antithrombin is going to decrease the amount of coagulation that occurs, right? If I'm antithrombin, I'm decreasing the amount of thrombin, which means decreasing the amount of coagulation. What antithrombin does is that it binds to coagulation factors and it prevents the coagulation factors from clotting the blood. Okay, I know that's kind of hard because I think it's new to you guys as first years, but the main idea is that this purple thing over here, this factor 10, promotes clot formation, and this blue box is going to bind to the purple thing. It's going to bind to the clotting factor to prevent clotting. Okay, if the purple thing is bound to something, it no longer can perform its action, and so it can therefore no longer perform the action of clotting specifically. Okay, so antithrombin prevents clot formations. What they want you to know is that heparan sulfate, using its NS domain, actually promotes the binding of antithrombin to coagulation factors. In other words, it promotes anticoagulation, right? Because I'm promoting the action of antithrombin. I'm allowing the antithrombin to bind to the factor and prevent the action of the factor. I know that's a lot of new things going on. Does that make sense at the very least how heparan sulfate is going to be an anticoagulant?
Yes.
Yeah? Okay.
Can you, I mean, I'm so sorry, but like I kind of like it's new to me, so like I'm kind of struggling to understand this slide. So can you repeat it, please?
Okay, so you understand where the heparan sulfate is, right? It's this thing over here, right?
Yes.
And we know that it has a domain, the NS domain, right?
Yes.
Okay, so just ignore that for a second, but I just want you to be comfortable with that. Just ignore that for a second. Focus over here. Okay, focus on these two things. Factor 10A is a coagulation factor. When you hear coagulation factor, I think I need you to think coagulation factor promotes clots. If I have more coagulation factors, it means I have more clot formation. So this purple one, coagulation factor, it promotes clots, okay? That's one main concept that I need to be clear. The second thing that I need you to know is that if this coagulation factor gets blocked, if something blocks the coagulation factor, it cannot promote clots. Is that clear until now?
Yes.
Okay. And the other thing that I need you to know is that one of the things that can block the coagulation factor is this thing over here, which is called antithrombin. Antithrombin can block coagulation factors. Okay, if it binds over here, it's going to block the coagulation factor. Okay, so you tell me, if antithrombin binds to the coagulation factor, is there going to be more clotting or less clotting?
Less.
Less clotting. Okay, perfect. So now that you understand that concept, the only new thing here is that antithrombin is like promoted, or its activity is increased if it binds to the NS domain. Okay, so the antithrombin is more likely to bind or block my coagulation factor if it binds to the NS domain of heparan sulfate. Does that make sense, the connection between this? I have heparan sulfate.
So one of the so like the heparin sulfate, basically it helps us in blocking the like aggregation or like clogging. So to prevent clogging, we use heparin sulfate, which is the NS domain, which it binds to the factor 10A, and it also increases the it increases the activity of AT, right?
So the NS domain binds to the AT, increases activity. And so now because the activity of AT is increased, the whole point of AT, the whole point of antithrombin is to block coagulation factors. So my NS domain isn't touching my coagulation factor directly. It's just promoting the action of AT. And the action of AT is to block coagulation factors.
Oh,
Does
okay,
that
I
make
get
sense?
it. Yeah.
Okay, so that's one way that heparin sulfate is an anticoagulant. That's one of the functions. The second one is much easier to understand, I think. So again, I have my heparin sulfate, and I told you that the heparin sulfate has this domain over here, which is called the NS domain. And the NS domain is very high in sulfur. So instead of binding to antithrombin, the NS domain can also bind to enzymes. I can also promote the activity of enzymes, okay? The specific enzyme that they're giving you as an example over here is called lipoprotein lipase. You can get from the name, but lipase, ACE is an enzyme, lip means fat. So lipoprotein lipase is an enzyme that breaks down fat, okay? So what's happening over here is that my lipoprotein lipase is binding to the NS domain. Once the lipoprotein lipase binds to the NS domain, it's going to increase the activity of the enzyme, which in this case means that there's going to be more breakdown of fats, okay? Because the point of lipoprotein lipase is to break down fats. So because I'm upregulating the enzyme, I'm breaking down more fats, and because I'm breaking down more fats, some of the fats can go into the cell and be used as storage. Okay, this part over here is the cell. Okay, does that make sense, or do you want me to repeat it?
Can you please repeat it?
Yes.
Okay, so we understand that this is my heparin sulfate, right? And we understand the domain. This is clear?
Yes.
Okay, and this blue one over here are my enzymes, okay? Okay, I'm just gonna explain the general concept first. The general concept is that if my enzymes bind to the domain, they're going to increase their activity. That is the main concept. If I have enzymes and they bind to this domain, it's going to increase the activity of the enzymes. Okay, is that much clear?
Yes.
Now I'm going to tell you an example of this. I'm going to give a specific example of an enzyme called lipoprotein lipase. What does lipoprotein lipase do? What does it break down?
Lipids or fat.
Exactly, it breaks down lipids. Once the lipoprotein lipase breaks down lipids, okay, the smaller fat components, like the lipids that had just been broken down, are going to diffuse across the cell membrane, and they're going to be used as energy from the cell. Okay, so my cell cannot use lipids for energy until they've been broken down, and they are being broken down by the enzyme lipoprotein lipase. Okay, does that make sense? What lipoprotein lipase does?
Yes.
Breaks down fat, and then once the fat is broken down, it can diffuse across the cell and be used as energy.
Okay, thank
Sorry,
you.
that means the positive heparin sulfate is lipid.
The what is lipid, sorry?
Part of the heparin sulfate is a lipid.
The heparin sulfate is not lipid. No, remember we actually went over the structure of heparin
Sorry,
sulfate.
why are they attached to it? Why are they attached to it?
Why does it
The lipid
upregulate?
region.
Like, why does it increase the activity? Sorry, I went too far.
No, why they attached to the domain of the heparin sulfate if they will not break it?
So, if they're not lipid,
Sorry, lipoprotein lipase is a breaking the lipid, right?
yeah, lipoprotein lipase breaks down lipids, yeah.
Yes, and they attach to the domain of the heparin sulfate.
The
What's the
enzyme,
function of it?
yeah.
Yeah, why they attach to the heparin sulfate? What's the function? That's my question.
The reason that the enzymes are attaching to the domain is because if the enzymes are just over here and they're not attached to the domain, they work slower. But if my enzymes are attached to the domain, they're going to work faster. The heparin sulfate helps the enzymes to work faster.
Oh, clear. Thank you.
Okay, so now I'm going to quiz you guys on the previous slide. So we went over this entire thing. We talked about heparin sulfate, talked about how it has a domain called the NS domain. And we talked about how ultimately it's going to prevent the action of factor 10A. What process does that prevent? What is the name of that process? I don't know.
Coagulation.
Exactly. You're preventing clotting. So that's how heparin sulfate acts as an anticoagulant. The second thing over here is that we talked about how another thing that heparin sulfate can do using its domain is that it can upregulate. Upregulate means increase the activity of certain enzymes. What is an example of an enzyme that can be upregulated?
Lipoprotein lipase.
Exactly, glycoprotein lipase. So these are two different functions of heparin sulfate. Okay, so now we've talked about the formation of glycosaminoglycans. The thing is, everything in our body needs to be recycled. Okay, so when things are formed, they also need to be degraded inside of our body. Okay, it's just the natural process that occurs. So what they're showing you over here is that I have, they're showing you kind of a general image, but let's just pretend these are my glycosaminoglycans, right? And what happens is that the glycosaminoglycans are going to be brought into the cell, and they're going to form this structure over here, which is called a vesicle. Okay, so my glycosaminoglycans are going to enter into a cell, and they're going to enter this structure over here, which is called a vesicle. My vesicle is going to fuse with something called a lysosome. Okay, and a lysosome is an organelle that is responsible for breaking down anything. Honestly, it's responsible for breaking down organic compounds. So I have my vesicle that contains my glycosaminoglycans, and it's going to fuse with my lysosome. Okay, and once that fusion happens, the lysosome is going to break down the glycosaminoglycans. You see how the glycosaminoglycans aren't being used anymore? And this is natural. Our body is supposed to be doing this. It's supposed to be forming things and then also degrading the things and recycling them. Okay. Is that much clear so far?
Yes.
Yeah. Okay. The other thing that they just want you to know about is that lysosomes use enzymes called acid hydrolysis to break down the GAGs. Okay, so I told you that lysosomes break down glycosaminoglycans, but I didn't tell you how. The lysosomes have certain enzymes, and those enzymes are called acid hydrolases, and those acid hydrolases are going to break down the GAGs. Okay, I hope everything is clear so far, because what we're going to go into is if there is a problem in this process. If for whatever reason my lysosome cannot break down the GAGs, I'm going to have an accumulation of the GAGs, right? That makes sense. If I have a problem with my lysosome cannot break down the GAGs, and so the GAGs are going to build up inside of my body, right? And those diseases that have a problem with the lysosomes are called mucopolysaccharidosis. Okay, so there's a group of diseases, and there are a bunch of diseases that have some problem with the lysosomes. And because the lysosomes aren't working, the lysosomes cannot break down the GAGs, and it's going to lead to an accumulation of the GAGs inside of my body. These are very, very severe, lethal diseases. Some of them have like very short or very poor prognosis, which means that the quality of life for these patients is very poor. So that's what they mean over here by saying it's progressively debilitating. They get even worse over time. Okay, so now we're going to give an example or talk about the mucopolysaccharidosis. So just to remind you guys, mucopolysaccharidosis are a group of a bunch of disorders that have a problem with the lysosome or the lysosome enzyme specifically. And because the lysosomes are not working properly, I cannot break down my GAGs. And because I cannot break down my GAGs, it leads to an accumulation of the GAGs inside of my body. Okay. So that's the main thing that they're trying to communicate to you over here. They give you an example of, or they give you two examples of a mucopolysaccharidosis. So the first example over here is called Hunter syndrome, and it's mucopolysaccharidosis type 2. And so let's just pause for a minute and let's just look at this. Okay, let's look at this entire thing. Let's orient ourselves. Over here, I can see I have my GAG. Remember, you guys know about this. You know glucuronic acid, you know N-acetylglucosamine. So I can see I have my GAG chain over here. And we talked about how some of the GAGs have sulfates that stick out of them, right? Sometimes my GAGs are going to be sulfated, okay? There's a specific enzyme called iduronate sulfatase that is responsible for removing the sulfate from my hyaluronic acid, okay? And I need to remove the sulfur from my hyaluronic acid to break down the rest of this GAG chain. Okay, so I need, one of the first steps in breaking down the GAG chain is that I need to remove this sulfur. If I can't remove this sulfur, then I cannot break down the rest of this chain. Okay, and there's a specific enzyme called iduronate sulfatase, which is responsible for removing the sulfur from the iduronate so that the rest of the breakdown process can occur. Okay, so some people are not going to have this enzyme, the iduronate sulfatase, and because they do not have iduronate sulfatase, they cannot remove the sulfur from the hyaluronic acid, and so they cannot break down the GAG. The GAG is going to accumulate inside of their body. Okay, that's like one of the main concepts. I just want to show you, I think I added an image, or maybe I forgot. Okay, I think, yeah, I have it over here. I just want to show you the sulfate attached to the hyaluronic acid, or where the sulfates are. So for example, like over here, this is my dermatan sulfate. Remember, this is an example of a GAG. And you can see I have my sulfate attached over here. And then we also went over this before, the heparin sulfate also has a sulfate group. So what I'm trying to convey to you is that there needs to be this enzyme called iduronate sulfatase that is responsible for removing the sulfur group, like over here and over here. Specifically for the two GAGs, dermatan sulfate and heparin sulfate. So I'm going to say this again, okay? Dermatan sulfate and heparin sulfate need a specific enzyme called iduronate sulfatase, which is responsible for removing the sulfur group, okay? And if I do not have this enzyme, I'm not able to remove the sulfur group, and then therefore I'm not able to break down the GAG chain. Okay, so that's why it says over here that the Hunter syndrome is that specifically dermatan sulfate and heparin sulfate are going to be affected because this specific enzyme, iduronate sulfatase, specifically acts on dermatan sulfate and heparin sulfate, okay? So that's one example of a lysosomal storage disease. Another example is this one over here called Hurler syndrome, okay? And this time, the enzyme that's deficient is called alpha L iduronidase. Okay, if you attended my talk yesterday, you'll know why it's called alpha, you'll know why it's called L. But pretty much what it's telling you is that this enzyme is responsible for breaking off this bond over here. Okay, alpha L iduronidase is responsible for removing the hyaluronic acid from the chain. Okay, so I just want to be clear in the difference. The first one, iduronate sulfatase, was responsible for removing the sulfur. However, alpha L iduronidase is responsible for removing the entire GAG from the chain, the iduronate acid. Okay, but once again, the same two GAGs are going to be affected in both of these conditions. It's still going to be dermatan sulfate and heparin sulfate, okay, because these are the two GAGs that have hyaluronic acid. The rest of my GAGs don't have hyaluronic acid, and so they do not need any of these two enzymes, okay? Is that clear what these enzymes do? Okay, I see some yes. That's good. I want you to remember these two, dermatan sulfate and heparin sulfate. I want you to remember that those two GAGs are specifically being affected by these two enzymes. So that's why I really emphasized on them, and I am going to quiz you on it later. Actually, I'm going to quiz you on it right now. What were the two GAGs that I just talked about that are affected if there's a deficiency in either one of these two enzymes?
Heparin sulfate.
Dermatan and heparin sulfate.
Dermatan sulfate and heparin sulfate, perfect. Okay, and then this enzyme over here, the sulfatase, what does it do?
It
It
removes
removes
the sulfate.
the sulfate from the
Hyaluronic acid, exactly, yeah.
yeah.
And then what about in Hurler syndrome?
It removes the gag from the chain, like your iodonic acid from the gag chain.
Exactly, perfect. Okay. Okay, and then they just give you two other examples that are also enzyme deficiencies, but just for different enzymes. But the overall effect is going to be the same. There's two other conditions. They also lack a certain enzyme, specifically a lysosomal enzyme that is supposed to be degrading the GAGs. And because people with these conditions don't have those enzymes, they cannot degrade the GAGs. That is like the main concept that's going on over here. I just showed you an example. I honestly don't know how much of this they're expecting you to memorize. I bolded the parts that I think you should pay attention to at least. I think you should at least know that Hurler syndrome and Mercois A have corneal clouding, because that was something that he specified on the previous slide over here. Like he did add an image of corneal clouding. So I would personally know if I was going to this exam, I would know that specifically Hurler and specifically Mercois A have corneal clouding. And the other thing that I would mention, again, because it's something that they like to test on in MCQs, is that the only mucopolysaccharidosis that is not autosomal recessive is Hunter syndrome. Hunter syndrome is not autosomal recessive. Instead, it's X-linked. So it's sex-linked, which means it's going to be more prevalent in males. Those are honestly the main two things that I would focus on. I would know the first and the last have corneal clouding, and I would know that Hunter syndrome is an X-linked inheritance, and that all of the other ones are autosomal recessive, because it's easy for them to ask them about it in MCQ. Okay, you don't need to know this. This was just an image that I added in case you guys are curious, but it's just in case you're interested in what these enzymes do. So for example, heparin sulfatase is responsible for removing the sulfur group from the heparin, right? And so again, if I can't remove the sulfur group, I can't break down this chain, and so there's going to be an accumulation of glycosaminoglycans. Do you guys remember which GAG I talked about in the very beginning that does not have an acid, but instead has galactose? Does anybody remember what the name of that GAG was?
The keratin one?
Yes. Yeah, good job. So the keratin, instead of having an acid, it has galactose. So it should make sense that if I lack this enzyme that is responsible or that acts on galactose, one of the GAGs that is going to be affected is specifically keratin, because keratin has galactose instead of the acid. So that's just an example I showed you over here. You can see galactose over here. This is the keratin sulfate. You can see galactose. You can see that galactose has sulfur. So the role of this enzyme, galactose 6 sulfatase, is to remove this sulfur. group over here. If I can't remove the sulfur group, I can't break down the GAG, and it leads to the accumulation. Similarly, over here, you can see chondroitin sulfate. You can see I have a galactose and acetylgalactosamine. It also has a sulfur group. I need to remove the sulfur group in order to break down the GAG. If I can't break down the GAG, it leads to an accumulation. That's what leads to mucopolysaccharidosis. That's the main concept. Again, you don't need to know the specifics. I just want you guys to understand what's happening. Okay, so now we are moving on to another concept. We are going to talk about glycosylation of proteins. So this entire time, what we've been talking about was proteoglycans, right? I want you to shift your head for a second and stop thinking about proteoglycans. Instead, think of glycoproteins. Okay, so we're shifting. We're not talking about, I'm done with proteoglycans. I'm now talking about glycoproteins. Glycoproteins are proteins. Does that mean that they have more protein or more carb?
Proteins.
Protein, right? So they're mostly protein. Here on the bottom over here is an example of a glycoprotein. So this green part is the protein part, and that you can see that attached to the protein, I'm going to add a sugar or a polysaccharide chain. So again, this is a glycoprotein. It's mostly protein, which is this green part over here, and there's going to be a small amount of sugar. Okay, so now we're going to talk about how does the protein attach to the sugar. Okay, and again, forget everything that you've talked about before. We're talking about a new concept. We're talking about glycoproteins. So it has its own set of rules. Okay, so I need you to be paying attention. There's two ways that a protein can attach to a sugar. One is called N-linked glycosylation, and another one is called O-linked glycosylation. What that means, N-linked glycosylation, means that I'm adding a sugar, glycosylating means adding a sugar to nitrogen, specifically N. And you can see that over here. You see this N over here, and it's being attached to a sugar. So sugar is specifically attaching to nitrogen, and I call that N-linked glycosylation, okay? The other form of glycosylation I have is called O-linked glycosylation. O stands for oxygen. So this time I have an oxygen, and my oxygen is being glycosylated. In other words, my oxygen is being added, or is having a sugar being added to it. Okay, so N-linked is over here, attaches to a nitrogen. O-linked is over here, and it means that the sugar attaches to an oxygen. Okay, here's what you need to know. Here are the rules of N-linked glycosylation. Remember that a protein is made up of amino acids, right? And there's like 20 or even more amino acids, right? One of the amino acids is called asparagine. Okay, I want you to look at this amino acid. I don't know how comfortable you guys are with the structures of amino acids, but they have a central carbon, they have a hydrogen, they have a carboxyl group, they have an amine group, and at the top over here, they have what we call an R group. An R group means that it can be anything. Okay, so this part of the amino acid always stays constant. An amino acid always has a carbon, always has a hydrogen, always has a carboxyl, amino, whatever. This part always stays constant. But one part of the amino acid is called the R group, and the R group changes between different amino acids. So what I want you to notice is that the R group, which is this group over here, of asparagine, which is an amino acid, has nitrogen. You see that over there? It has a nitrogen, okay? And the presence of this nitrogen is what allows for N-linked glycosylation, because the sugars are going to start attaching to this nitrogen over here that is in the R group, okay? So if I have N-linked glycosylation, it must mean that I must have an asparagine, because asparagine has an R group with an N, and I need that N to do N-linked glycosylation. Okay, so N-linked glycosylation can only happen if an asparagine is present. That is what is so important. O-linked glycosylation, on the other hand, can happen using two different amino acids. Okay, so again, let's look at our amino acid. I have my central carbon, I have my carboxyl group, my amine group, my hydrogen, and I have this segment over here, which is my R group, right? And if you notice in my R group, I have this oxygen right over here. This oxygen is what is going to be glycosylated. In other words, this oxygen is going to attach to a sugar, okay? And once that oxygen attaches to the sugar, I call that O-linked glycosylation, okay? That's what happens if there's a serine. Similarly, I can do the same thing if I have a threonine. So look at my threonine, I have my central carbon, amine group, carboxyl group, H group, and then I have this group over here, which is called my R group. Okay, if I'm looking at my R group, I notice I have an oxygen. This oxygen can attach to a sugar. Once that oxygen attaches to a sugar, it's going to be called O-linked glycosylation. So the main concept that I really want you guys to understand, N-linked glycosylation needs asparagine. O-linked glycosylation needs serine or threonine. Okay, but I need serine or threonine to do O-linked glycosylation, and I need asparagine to do N-linked glycosylation. Okay, and it's because of the R groups that they have. I am going to quiz you guys on this. So we talked about what O-linked glycosylation means. We talked about what N-linked glycosylation means. For N-linked glycosylation, what is the name of the amino acid that needs to be present?
Asparagine.
Asparagine, perfect. And what about O-linked glycosylation? What two amino acids can I have?
Serine and threonine.
Perfect. Okay, so that's really important. It's one of the main concepts of this. Okay, this next one, I don't know how high yield it is, but there are three main structures that I want to talk about. I honestly don't know why they want you to know this, but they want you to know about three things. They want you to know about fibronectin. So focus over here. They want you to know about this structure over here called integrin. And they want you to know about one more called laminin. Okay, so focus on these three molecules, for example. Let's start with fibronectin. What they want you to know about fibronectin, so fibronectin is over here, okay? What they want you to know about fibronectin is that fibronectin can attach to all forms of extracellular matrix except type 4. So type 1 extracellular matrix, type 2 extracellular matrix, type 3 extracellular matrix can all bind to fibronectin, but not type 4, okay? That's what they want you to know. So we're looking at the extracellular matrix. You can see by fibronectin, and I can see it can attach to different types of extracellular matrix, but only 3 types, okay? Type 1, type 2, type 3, cannot attach to type 4. The other thing that they want you to know about is this structure over here called integrin, right? You can see the thing that it's pointing at, integrin. And if you hear about a structure that is transmembrane, the first thing that should pop into your head is integrin. So the main thing you need to know about integrin is that it is a transmembrane molecule. Transmembrane means it's going across the length of the membrane. Okay, so integrin is transmembrane. The last thing they want you to know about is laminin. Remember how I told you that fibronectin can only attach to extracellular matrix type 1, 2, and 3? Well, the structure that's going to attach to extracellular matrix or collagen type 4 is actually called laminin. Okay, so laminin attaches to collagen type 4, and you find collagen type 4 in what we call the basement membrane. Okay, so if I have my membrane over here, there is something that sits right below it, and we call it my basement membrane or my basal lamina, I think some people call it. And my basal lamina has type 4 collagen, and you need to know that type 4 collagen attaches to laminin. Okay, I'm going to quiz you guys on this on the next slide. Okay, so reading the first one, what is the glycoprotein that attaches to all types of extracellular matrix except type 4?
Fibronectin.
Perfect. Okay, and then what is the structure that does attach to type 4 collagen?
Excuse me?
Laminin.
Excuse me?
Laminin.
Yeah?
Excuse me, can you explain to me the glycoproteins? Because I think my iPad turned off as you were explaining it.
Like what a glycoprotein is?
Yes.
Okay, so a glycoprotein is a protein, right? So this whole black part, this whole green part, sorry. Okay, so this whole part is a protein that's attached to a small amount of sugar. So glycoprotein means mostly protein with a small amount of sugar, right? So this is the protein, and I have a small amount of sugar, and we call this entire structure a glycoprotein. Is that clear?
Yeah.
Okay, were you here for the N-linked and the O-linked?
After like the diseases and like the deficiencies, my iPad turned off. So like after the monocytitis or something like that, my iPad turned off.
Oh, okay, so you don't know this part.
Yeah.
Okay, so I'll try to go over it quickly, but the main concept is that if I have a protein, okay, proteins are made up of amino acids, right? And one of the amino acids that can be present is an amino acid called asparagine, okay? And asparagine has a nitrogen, okay? You can see this nitrogen. This nitrogen is responsible for attaching to sugars. So the main concept that I'm trying to get out here is that if you have N-linked glycosylation, which means a sugar being attached to a nitrogen, you need the amino acid asparagine to be present because asparagine has nitrogen. So only if asparagine is present can N-linked glycosylation occur, because we need the nitrogen of asparagine for N-linked glycosylation occur. So attached to this nitrogen is going to be a sugar. So you can see the nitrogen over here, and you can see the sugar over here. So the sugar specifically attaches to the nitrogen of asparagine. Okay, that's it for N-linked glycosylation. So for O-linked glycosylation, it's the same concept, except this time, instead of asparagine, you need either serine or threonine. Okay, the reason you need serine or threonine is because serine and threonine has an oxygen, and this oxygen is needed for O means oxygen, O-linked glycosylation. In other words, this oxygen is what attaches to the sugar. Okay, so I can only have O-linked glycosylation if I have serine or threonine, because serine and threonine have oxygens, and these oxygens are needed for the attachment to sugars. Okay, for O-linked glycosylation. Is that clear?
Yes.
Okay, and then we just revise these concepts. So do you want to answer it really quickly? What's the amino acid that's associated with N-glycosylation?
Asparagine?
Yes. And then what are the two amino acids that can be used for O-glycosylation?
Serine and threonine.
Perfect. Okay, and then I pretty much just talked about over here. It's a very simple concept. All you need to know is that there are 3 glycoproteins that they want you to know about. One is fibronectin, one is integrin, and one is laminin. Okay, the role of fibronectin is to attach to extracellular matrix type 1, 2, 3, but not 4. Okay, fibronectin attaches to extracellular matrix type 1, 2, 3, but not 4. Okay, that's the main concept of fibronectin. That's its role. It's just a structural role. For integrin, the main thing that they want you to know about is that it's transmembrane, meaning it's going to be placed inside of the membrane, or it's integrated inside of the membrane. That's why it's called integrin. Okay, and then the last one is laminin, which is a glycoprotein that attaches to collagen type 4. Remember, I told you fibronectin does not attach to collagen type 4 or extracellular matrix type 4.
So basically, fibronectin, it attaches extracellular matrix to the cell membrane, or where to what exactly?
Yeah, to the cell membrane.
Uh-huh.
Exactly.
So fibronectin, it connects extracellular matrix 1, 2, and 3, and then we have the laminin that connects the fibroblast 4 to the cell membrane, and then we have the integrin, which is basically at the cell membrane, or it's at the transmembrane.
Exactly. So those are the main
All right.
concepts you need to know. So that was the same thing that we were just going over right now. Do you want to answer them really quickly? Which one is the one that does not attach to type 4?
The fibronectin.
Yeah. Which one does attach to type 4?
Wait, hold on, let me remember. So we have one first fibronectin, and then we have the it's called the laminin, and then the integrant,
Which is?
which it's basically acts on the transmembrane.
Exactly. It's transmembrane. Yeah. So that's the main concept. And the only last thing that I talked about is that type 4 collagen is found in the basement membrane, or what's also called the basal lamina.
Yeah.
That's the
All right. And I think we took already the O-linked glycylation and N-linked glycylation.
O-linked and N-linked. Yeah,
Yeah.
I covered that in the previous slide. You're right. Let me just see how
Yeah.
much. Okay.
I think we're done.
Pretty much, yeah. This is just revision. So all that it's telling you is that this is the O-linked glycosylation. I told you that O-linked can either be serine or threonine, because those are the amino acids that have oxygen.
And N-linked is Agrican.
Is asparagine.
Asparagine, right.
Yeah, asparagine, exactly. It's right over there. The only new concept is that they're telling you is that, so we talked about serine and threonine, right, as the amino acid. The sugar that they can attach to for O-linked glycosylation can either be mannose or it can be N-acetylgalactosamine. Okay, so the sugar that is going to be attached in O-linked glycosylation is either going to be mannose or it's going to be N-acetylgalactosamine. Okay, the reason I'm emphasizing that is because if I move over to N-linked glycosylation, the sugar can also be mannose, but it's going to be N-acetylglucosamine. Okay, so N-acetylgalactosamine is the sugar in O-linked glycosylation, and N-acetylglucosamine is the sugar in N-linked glycosylation.
Yeah.
Okay, that's also important to know. And then I think they also want you to know what dolicol is. So I'll show you dolicol on the next slide. Dolicol is just this protein over here that is specifically required for N-linked. Okay, not for O-linked. It's dolicol is specific for N-linked glycosylation, and it just helps with the process. It just allows for the transfer of the sugars onto the protein. Okay, so the structure that transfers the sugars onto the protein is called dolicol, and it's specific for N-linked glycosylation. Okay, it's not relevant in O-linked glycosylation. Okay, one just last thing, okay, is that there's a condition called eye cell disease. Okay, before I talk about the disease, I'm just going to briefly go over normal physiology. Remember how I told you guys, everything in our body needs to be degraded. As part of natural life, things need to be degraded, okay? Glycoproteins are not exempt of that, okay? Glycoproteins also need to be degraded. So what happens is that when a glycoprotein needs to be degraded, it's going to be tagged with a phosphate onto the mannose sugar. Okay, so look, this is my glycoprotein. If my body senses that this glycoprotein needs to be degraded, it's going to add a phosphate onto the mannose. Okay, there's going to be a receptor called the mannose 6-phosphate receptor, which is going to sense the phosphate, and it's going to signal that this molecule that has been tagged needs to go to a lysosome to be degraded. Okay, so the main concept is that part of normal physiology is that our body is going to tag glycoproteins that need to be degraded with a phosphate on the mannose, and that there's a receptor that is going to sense that phosphorylated mannose or that mannose 6-phosphate. And if that receptor senses the mannose 6-phosphate, it's going to signal to your body that this glycoprotein needs to be degraded. Okay, so remember how we talked about before that we have these mucopolysaccharidosis, right? Which was when GAGs cannot be broken down, right? Now we're talking about a similar concept, except instead of GAGs, now we're talking about glycoproteins. Okay, now we're talking about the breakdown of glycoproteins. So what happens is that there is a condition where I cannot phosphorylate this mannose. If I cannot phosphorylate this mannose, the mannose is never going to be tagged. It's never going to be sensed by the receptor. And that means that this glycoprotein is never going to be degraded because it was never phosphorylated. If the glycoprotein is never going to be degraded, it's going to lead an accumulation of the glycoprotein. And we call this condition eye cell disease. So eye cell disease is a deficiency in the ability to phosphorylate mannose, which ultimately means that eye cell disease is ultimately going to lead to a lack of degradation of glycoproteins, which eventually means I'm going to have an accumulation of glycoproteins in the body. Is that clear?
It is accumulation of glycoproteins.
Yes, accumulation of glycoproteins in the body because they're not being broken down. Okay, that's it. Is there any questions? I hope it was helpful.
Nope, thank you.
Okay.
We're done or we're gonna solve questions?
No, we're done.
Thank you.
Thank you.
And I'm sorry, I think I took a lot of time with you.
No, it's okay. It's your right. That's what these sessions are for. They're to learn. I hope it was helpful.
Yeah. Okay, but like I'm gonna have to like rewatch the recording since like a lot of I was not able like to catch some stuff because my iPad turned off and stuff.
Oh, okay.
Bye.
That's okay. I think they'll have the recording up soon.